Hello and welcome to IB Biology Notes with Dr. Kimmo. This is A3.1 Diversity of Organisms. This subchapter is about variation between organisms, between species, what we actually mean with the word species. It's about chromosome numbers, genome size, and in higher level, it's also about some modern technology. Life comes in many shapes and forms. Variation is a defining feature of life. There is variation between phyla, between species, between family members, even between identical twins. Throughout history, in all cultures, people have recognized that every organism is a member of a group of similar individuals, a species. Species have obviously been named in a local language. So for example, this bird here, some people would call it a barn swallow, some would say rauchschwalbe, others harapääsky, golondrina común, and many other things which I'm not able to pronounce. Carl Linnaeus introduced the binomial naming system. He came up with this system where every species gets a Latin name, and that makes it easier for people from different language groups to talk about the same species with the same Latin name. This bird would be Hironda rustica, and the first name, Hironda, is the genus of this bird, the genus where this bird belongs. The second name, rustica, is the species, and there are always two names. That's why it's called the binomial naming system. It's very useful to have an international naming system to keep track of all the species, because more than 2 million species have been classified or named so far. The actual number of species is believed to be much higher, approximately 8.7 million. This is an estimate, and whatever the real number is, that number is believed to decrease in the future due to species extinction. From this graph, you can see that almost half of the named species are insects, and the second largest group is flowering plants. You can find mammals almost at the bottom of this chart with less than 7,000 named species. The pie chart on the right shows that almost 90% of all named species are animals. 7% are fungi, 3% plants, 1% protozoa. Then we have chromists, bacteria, and archaea. It says 0%, but that is rounded down to 0. So, of course, it's more than 0. It's 0 point something. All right, so more than 2 million listed species. But what is a species really? There are many definitions, and the most common and accepted biological species concept says that it's a group of organisms that can interbreed and produce fertile offspring. However, this definition may lead to conclusions that are not generally accepted. For example, a mule is a hybrid between a male donkey and a female horse. It's almost always infertile. So here the species concept works pretty well. You have horses, you have donkeys. Well, they can have mules, but the mule cannot have offspring. A liger is the hybrid between a male lion and a female tiger. Female ligers are sometimes fertile. Still, we don't consider lions and tigers as belonging to the same species. We see them as separate species, although they can in some cases have fertile offspring. Several individuals of the same species form a population. The definition is a group of organisms of the same species living in the same area at the same time. A population may grow in size, it may spread over a larger area and eventually split in two. Then there might not be any interbreeding between these two new subpopulations, but we would still consider them as belonging to the same species. However, as time passes, these two subpopulations will adapt to their own local environment and will diverge, which means they will develop in different directions. Eventually, this leads to speciation. So instead of two subpopulations, we have two species. It's a long process, and the question is, at which point should these two subpopulations be considered as separate species? The image on the left shows bear species of the genus Ursus. They are generally geographically isolated from one another, which means they live in different areas. There may be hybrids, however, and hybrids between some species may be fertile. So this does not really fit with the biological species concept either. Chromosome number is a fundamental characteristic of a species. Each species has a certain chromosome number, and in plants and animals the number is usually even. For example, human somatic cells have 46 chromosomes, and a somatic cell is any cell except for a gamete. Human gametes have half that number, 23. And it makes sense if you consider that a sperm cell and an egg cell fuse to form the zygote, and they each contribute with 23 chromosomes, so the zygote has 46 chromosomes. The number of chromosomes in a species is not that relevant. It does not reflect, for example, complexity. Have a look at the table on the right and you will see that wheat has almost the same number of chromosomes as humans do, and potatoes have even more. The number of chromosomes can change during evolution by fusion or splitting. For example, chimpanzees have 48 chromosomes, whereas humans have 46, which means they have one more pair than we do. And that's what their and our shared ancestor also had. It had 48 chromosomes, and at some point in the human lineage, two of the chromosomes fused to form one chromosome. Karyograms can reveal chromosomal abnormalities. A karyogram is what you see here on the right, a graphical presentation of a karyotype. A karyotype is a set of chromosomes, and karyotyping is examining a set of chromosomes. To do this, mitosis is halted when chromosomes are condensed at metaphase. And at metaphase, the chromosomes actually have two sister chromatids, but usually only one of the two sister chromatids is shown in a karyogram like this. The nuclear membrane is broken after mitosis has been halted, and the chromosomes are stained to create this banding pattern of black and white stripes. This makes it easier to distinguish one chromosome from another. The chromosomes are then photographed and the images are arranged into a karyogram. And a reason why a doctor, a scientist would do this kind of karyogram would be to find out, for example, whether a person has trisomy. Trisomy 21 is when a person has 3 copies of chromosome 21, and that leads to Down syndrome. This karyogram comes from a person who is male. You can see XY at the bottom right. If this person were female, then she would have 2 X chromosomes. So XY is male, XX is female. A genome is all the genetic information of an individual, a population, or a species. We can talk about, for example, a person's genome or the entire human genome. The genome contains genes, and a gene is a DNA sequence which is transcribed into RNA and then usually translated into a polypeptide. But it's not always translated. Also, the DNA sequences which code for ribosomal RNA, for example, they are also genes, but they are not translated into a polypeptide. In all members of a species, a certain gene is always located in the same position, on the same chromosome, and we call that position a locus. On the right, you see two homologous chromosomes. They have the same loci, which is plural for locus. They have one locus for eye color, one for blood group, and one for earlobe type. And these are just examples. These loci are actually not on the same chromosome. Okay. So just an example. Now members can have different versions of a gene, and those different versions we call alleles. So in this case, a person with two homologous chromosomes, and everybody has two homologous chromosomes, on one of these chromosomes the person has an allele for brown eyes, on the other an allele for blue eyes. Whether the eyes actually become brown or blue is then determined by which allele is dominant over the other, or whether they are co-dominant. That's something you will learn later. In this specific case, actually brown eyes, the allele for brown eyes is dominant over the allele for blue eyes. So the eyes will be brown. Here's an allele for blood group A, and here's an allele for blood group B. It's the same gene, the same locus, but different alleles. And finally, here's an allele for free earlobes, and here's an allele for attached earlobes. So these different versions of a gene are called alleles. The base sequence is very similar in all of us, yet everybody is unique. Scientists have identified a few thousand positions where genes can differ with one base, and this leads to alleles. In each individual, only one in approximately 650,000 base pairs is different from the most common version. Human genetic diversity is higher in Africa than elsewhere, and this is in line with the out of Africa theory that humans developed in Africa and then some populations emigrated out of Africa and then spread around the world. Whatever differences there are between humans, they are based on very few differences at the genetic level. So we humans are largely similar. However, no two individuals are genetically identical, except identical twins. Let's zoom out from humans and consider different species. There is great variation between eukaryote genomes. Species with a recent common ancestor have a more similar base sequence than species with a distant common ancestor. And some genes differ more than other genes between species. On the right, you can see the cytochrome C oxidase protein complex embedded in a cell membrane. Now the base sequence encoding for this protein complex is similar in all eukaryotes. It's a protein complex involved in the electron transport chain of cellular respiration, and this is a very old mechanism, and it's similar in all cells with aerobic respiration. So you cannot expect big differences in the base sequence encoding for this protein complex in humans versus any other species with aerobic cell respiration. However, there are other proteins which can be very different between species. For example, proteins that are important for brain development. So the base sequence can be very different than between different species, humans versus any other species. Here is an example of the human insulin 1 pre-pro protein. It's part of the sequence in human versus mouse, and it's more than 80% identical between human and mouse. The lines between the letters show that humans and mice have the same base, and when there's no line, it's not the same base. Eukaryote genomes vary in size by a lot. Genome size does not always reflect species complexity or even the number of functional genes. So it's a bit like the number of chromosomes does not always reflect the complexity of a species. When you study the graph on the right, pay attention to the x-axis where the scale is logarithmic. Many species contain a lot of non-coding DNA, of which some have no known function. This DNA is called junk DNA, and it means that a species can have a lot of sort of extra DNA that, as far as we know, doesn't do anything. At least it does not code for any protein. So that can sort of enlarge the genome without increasing complexity. At the same time, some genes can produce several different types of proteins through what we call alternative RNA splicing. So one gene produces a pre-mRNA, an RNA molecule, and this RNA molecule can then be spliced into different segments, and that way the same gene can actually produce different proteins. So that's like the opposite from junk DNA. You can have relatively small amount of DNA, but then if you use the same DNA to produce different proteins, you can have more complexity with less base sequence. Reading the DNA base sequence of a gene is called sequencing. And reading or determining the DNA base sequence of an entire organism is called whole genome sequencing. Sequencing of short DNA fragments became common in the late 1970s when Sanger sequencing was developed. This method is still in use today, but mostly for screening of short sequences, like new variants of the COVID virus, for example. It's expensive and laborious on a large scale, but very practical for small fragments, very accessible, and it doesn't require large investments. On the bottom right, you see modern high-throughput sequencing equipment, much faster, much more expensive. Nowadays, large-scale sequencing of whole genomes are performed with fast, cheap, and automated high-throughput methods. The first organism to be fully sequenced was the bacterium Haemophilus influenza in 1995, and the entire human genome was sequenced in 2003. Whole genome sequencing has become increasingly fast and cheap. When you study the graph, pay attention to the Y scale, which is logarithmic. The white line shows Moore's law, which is known from computer science, and says that every 2 years the number of transistors on a chip doubles. So this is just for comparison. That's a very fast development, and during the first years shown in this graph, the price went down. Price for whole genome sequencing went down more or less according to Moore's law, which is just a coincidence. But then it suddenly became even more cheap, and In about 20 years, the price per genome sequenced has gone from about $100 million to less than $100. Whole genome sequencing has taught taxonomists a lot about phylogeny. It has revealed unexpected twists in human evolution, and it has allowed doctors to start developing personalized medicine. And we will have a closer look of each of these 3 points. Taxonomy. Taxonomists have been able to construct detailed phylogenetic trees based on similarities between genomes. The rooted tree shown here is based on whole genome sequencing of about 200 species from all 3 domains. Rooted means that it has a starting point, and the starting point is in the middle of this circle where it says LUCA, last universal common ancestor. And we can zoom in to find Homo sapiens here. You see that our closest relative is Pan troglodytes, and that is the chimp. Of course, this study is not based on all the genomes that have been sequenced, but about 200 species. Whole genome sequencing has also taught evolutionary biologists and anthropologists a lot about human ancestors, evolution, and migration. You know already that the human genome was sequenced about 20 years ago, and since then, the Neanderthal genome and the Denisovan genome have also been sequenced. Neanderthals lived mostly in Europe and Denisovans in Asia, and they both went extinct 30 to 40,000 years ago. However, based on whole genome sequencing, it seems that Europeans and Asians have 1.5 to 2% Neanderthal sequence in their genomes. Some populations in Oceania have up to 5% Denisovan sequence in their genomes, and this indicates that Neanderthals and Denisovans, which used to be seen as separate species from modern humans, actually interbred with modern humans, and they are now most often considered as members of the same species as modern humans. Neanderthals, for example, are usually classified as Homo sapiens neanderthalensis. Personalized medicine is an emerging field that shows great promise and has benefited a lot from whole genome sequencing. The idea is that medicines could be tailored for an individual based on their genome. Among the medicines we use today, some work for some people but not for others, and designer drugs tailored for individuals could solve this problem. That's the idea with personalized medicine. That's all for now. See you next time.